Um, Please note that the session will be recorded. Welcome everyone to today's webinar. I have Monika and Yvonne who will be presenting on the introduction to sequencing tech. I don't know who's going to start between Yvonne and Monika. Probably you guys can decide who will go first.
Sure. Yeah, I'm going to get started, or I'll start it off and then I'll hand it over to Monika.
All right, cool. So I guess we can proceed.
Great. Let's see. I'm going to share my slides, my screen here. So I'm Ivo Violich. I am not a doctor. I get put on as a doctor sometimes, but I don't have a PhD. I have a master's in bioinformatics. My background is in, I've worked a lot in sequencing cores and biobanks, and joined UCSC and HPRC about 6 months ago, working with Karen and Benedict. Yeah, so I'm the lead data wrangler on HPRC, and then I'm also doing quite a bit of work with Karen with the production center at UCSC for the HPRC, which is a ONT production center. Can you guys hear me okay? All right, it's very early, so I don't normally sound like this. I have my morning voice still. But we'll all be on the same, good, we'll all be on the same time zone soon. So let's see. So a quick overview. Let's see, 2 days ago, you guys heard from Karen and Benedict and got a great sort of overview of the HPRC project and the T2T. The goal of HPRC and sort of how it's structured and the high level why it's being done and all that. And then yesterday you got great presentations regarding sort of more details on the assemblies and the pangenomes, which are the products of the HPRC and the applications. So I'm gonna Monika and I are gonna go over. I may have left off a couple things here. I'm gonna try to sort of go over a little bit of the structure, the data structure, the data sphere, if you will, of HPRC, how it's organized, how it can be accessed, and then do a brief overview on PacBio HiFi, and then a little bit more in depth on ONT. And then I'll hand it over to Monika to talk about the algorithms and sort of the approaches of doing some of the same things that were discussed yesterday, but using just ONT technology. All right, so maybe I should go into slide presentation mode. There we go. So the HPRC data biosphere, you can think of it, I call it data biosphere because there is this sort of open data ethos around, or that's part of HPRC. So we want all the data to be very accessible, shared wherever people will use it, and try to keep costs down around that as well. So that's why I use that term. And it really is quite a collection of data. So we've talked about over here on the right, you can see the products. This is a representation of the pangenome down at the bottom. And then these are the phased assemblies that go into the pangenome. So those are obviously both available, but there's also this incredible collection of raw data that makes up the assemblies. So we have PacBio HiFi reads, Nanopore ultralong, Illumina trios. Those are taken from 1,000 genomes. So they're just integrated in. And then IC data to supplement the Illumina data if it's missing. So all that is organized and available, like I was saying, hopefully in everywhere that folks would could use it. So the data is mirrored in GCP buckets, so the Google Cloud, and in AWS S3. The GCP bucket is the backend, or it still is the backend for until the end of the year, for Terra. So if you're using Terra or Anvil, that's how you would access the data, the HPRC data. And then otherwise, you could access it via the AWS S3 bucket. And I've highlighted that one because that's the one that we mainly use because it is subsidized. So you can download that with no egress charges. We also try to have open GitHub repos for all the processes that go into not only the assemblies and the pangenomes, but also the data wrangling itself and the QC of that data. And then just for this to live on, we've also put it in the federal repositories, data repositories. So I'm mostly putting in SRA, but some of the other data types go into these other public data repos. And all this is very well laid out in this web page. So if you have more, if you want to look into the details of how the data is spread out and how to access it and get all the links, this is the website to go to, the humanpangenome.org, and then /data for data types. Any questions on that? That was just sort of a very brief overview there. Okay, so moving on, want to talk briefly about PacBio HiFi. You can think of it as Illumina-like quality, but with long reads. So I mostly worked with Illumina data, so short read data of 150 base pairs, paired end, so 300 base pairs max. Here we're talking about 15,000, 15 KB, and so very long reads. But the quality is the higher quality is achieved by creating the circularized DNA that's trapped in a little light well and just repeatedly sequenced. So you can see this. This is what this representation, this figure is representing. So this circular DNA is just sequenced over and over again. And so these are called subreads in PacBio speak, and all those subreads get sort of collapsed into a single HiFi read with very high accuracy. But the subreads themselves are lower accuracy. So here's a few sort of key things to remember about PacBio HiFi. The HiFi reads are produced using circular consensus sequencing, so CCS. I believe that's also the name of the program that creates that outputs the HiFi reads from the subreads, and that can be done on the sequencer. So the output is normally HiFi reads, and you can save sort of raw data in the form of subreads. The read length, as I mentioned, for PacBio HiFi is 15 to 20 KB. The quality is Q33. That's the Fred score. If you're not familiar with Fred score, that translates to, I think they said in the slide before, like 99.99% accuracy, or the likelihood of an error being 1 in 2,000. The outputs that you're sort of dealing with from PacBio HiFi data, or the format that you'd be using, is most likely unaligned BAMs or just FASTQ. The thing to remember about unaligned BAMs versus FASTQ is that in a BAM format, you can store a lot more information. So it's not just a sequence. The format is more flexible, so you can have tags that include, in this case, kinetics or modification. So and just more information than a FASTQ. The FASTQ format is very simple. It just has the sequence and the and the quality and a little bit of metadata. And so, I did not know how to work slides. That was just me double clicking. Apologies. For both of these technologies that I'm going to talk about today, PacBio and ONT, you're sequencing native DNA without amplification. And the reason that's important is that it allows the study of modifications because the modifications are still there. And when I mentioned kinetic information being stored in the unaligned BAM, and that's what would be the only reason that's possible is because it is native DNA. And I bring this up because there will be HiFi data as part of the workshop, and I think the main thing to check is that was kinetics information sort of stored or preserved and included in the base calling. Oh, here's a little bit on Fred scores. I think Benedict or Karen mentioned the Q100 project. So that was an effort to sequence to, Q100, which is, almost error-free. If Q60 is one in a million, the probability of error. Okay, so here's a little bit more on kinetic signatures. So I was mentioning that this is how the PacBio sequencing sort of works at a very high level. The piece of DNA is sort of trapped in this light well, and as each base is attached, you get a light signal, and that can be then translated to a sequence. But this light signal can be different based on if there's modifications or not. So that's this kinetic signature. And then that using a learning model, using a model that can be then translated to modifications. So one thing to know is that although you can't, and this isn't strictly the case, but for HiFi, you're not usually rebase calling the sequence. But if you store the raw kinetic data, you can recall the modifications because that is just this the kinetics of how the light pulses were different based on modification or not. Okay, and so here's a couple things on PacBio hardware and software. The data that we're going to be using for the workshop that already received is from a Revio, but I'll just mention this just in case. Google Health developed in the last couple of years a better base caller than PacBio had on their systems, and it's called Deep Consensus. And then very recently, in this summer for us, in the last few months, the Revio has rolled out. This is a new sequencer from PacBio that replaces the Sequel, most recently the Sequel 2 that was running. And Deep Consensus is the base caller on the Revio. So it's the output from the Revio is Deep Consensus called bases. We probably don't have to worry about it, but if you have Sequel data, you could rebase call it using Deep Consensus. But for now, we don't have to worry about this too much. Just know that the Revio is brand new, and folks are still sort of figuring out how to make sure that the data is retaining all the information that you need for future rebase calling or future modification recalling. Any questions on PacBio, sort of hardware and base calling and data types? All right. So now moving on to ONT. So ONT is different when compared to PacBio because it's, although it is sequencing native DNA, it's doing it through a pore. So this is a representation of that. So the DNA is going through this pore, and there's also electrical current going through. So the ONT wants you to think about this as, the electrical current is water sort of going through a pipe. The pore is the pipe, and the DNA is like you're putting a stick into the pipe and disrupting the flow of the water. And that's how you know sort of what bases are going through as the DNA molecule goes through. So in terms of ONT, the key aspects to remember are that it's a voltage signal. That's the data that's being stored in the POD5 format. So the raw output of the ONT sequencer is electrical signal. And these files are huge. These can be up to 1 terabyte per flow cell. And for HPRC, where the target is 30x coverage on 100 kilobase or more read length, we use 3 flow cells per sample, and sometimes 4. So that's 3 terabytes per sample that we're sort of storing for reasons that I'll go into. 2 of the ONT preparations and technologies that we'll be using is ultra long sequencing. That's read lengths of 100 to 300 kilobases, and duplex, which is shorter but more accurate. You can see that ultra long is at Q23, and duplex is starting to get closer to that Q30 quality. That's the sort of Illumina standard that HiFi can reach. The outputs, again, are unaligned BAMs or fast Qs. We'll most likely have unaligned BAMs. Just like with the HiFi data, you can store more information. So for duplex data, you can store whether the read was a duplex read or not. And you can also store modification information. Once again, you're sequencing native DNA with no amplification. So it allows for the study of it allows for the study of modifications. And for ONT, you're storing the raw, like I was saying, you're storing the raw voltage signal. So you can rebase call the sequence and the modifications if you retain this POD5 data. So a little bit on duplex. So up until recently, all ONT data was simplex. So a single strand of DNA would go through, and that would be stored in the POD5 data, and then you'd base call that, and you'd get, that's the read that you would get. Recently, they developed a new flow cell that would tether the DNA to the membrane so that after the first strand of the DNA would go through, the second strand would go through. And this allowed them to come up with this base calling algorithm that's stereo base calling that allows for the correction of any errors in that first pass, and increase the quality significantly. So now you're getting much higher quality. It does come at the expense of read length and throughput. So you get less data per flow cell, and your reads are shorter than the ultra long, but the quality is increased dramatically. And so your output is a BAM for duplex, and it has a DX tag. So this tag lets you know if the read is a duplex read, if it's a simplex read that did not create a duplex, or if it's a simplex read that created a duplex. So to create a duplex read, you need 2 simplex reads. So all these are stored in the same BAM, and then you do a split command with SAM tools to get a purely duplex read BAM, and then a purely simplex read BAM, and then this simplex that created duplex reads. So you get 3 BAMs out of it. And that's one thing to note, is that you do get some simplex data out of this. But again, it's not as long as ultra long. So briefly, the ONT hardware. This is the hardware that is on its way. It's a Promethean sequencer and server. The one thing I want to point out is that this tower here is a pretty beefy GPU tower. Has 4 GPUs in it, considerable storage and RAM. So you can do base calling directly on this tower. So this runs the sequencer, but it's also used for base calling, which is GPUs are necessary just to make it a quicker process. For ONT software, these are the sort of the ones to know. They're all named after fish to some extent. So Minnow is what controls the sequencer, generates the POB5 data, and can do live base calling. So you have the option of base calling as you're sequencing. And then the ONT base call client is how you would base call on the Promethean server after the run is complete. And it's sort of a mix of Guppy and Dorado, and it's a little bit difficult to use, but once it's not too bad. And then if you do want to base call off of the server, off of the Promethean server, you can use Guppy, that's the old base caller, or for newer data, you'd use Dorado. That's the current base caller. It's open source, which is the departure. Guppy, you used to have to sign up and download it from ONT. But there's a really great GitHub repo with Dorado in it, and that's great support. So this is how we would base call on outside compute or outside of the Promethean. For base calling, I think I'm going a little bit long, so I'll just speed up a little bit here. ONT uses neural networks for base calling. So you can see here, this is on the left is what you would, a representation of the electrical signal as the DNA molecules going through the pore. This converted to what they call squiggles, and then that's eventually converted to base called sequence. These rely on data that's been trained on known sequence, and these are the models that you give as input to base calling. And you have to, the models are constantly being updated and trained and improved. So the idea is that you sequence your data once and you keep seeing improvements because you can re-base call that data. This is a dated chart from ONT, but this is the same sequence data, and then over 2 years, they were able to increase the accuracy by rebase calling it on new base callers with new models. There is drawbacks to that. The POD5 data is very large, so you end up storing these huge files in hopes that a model will come along that will help you. And then choosing the right model can be a little bit tricky if you're just using new library, new kits and the most recent tech from ONT, then it's not too bad. But if you start trying to rebase call older data, that can be really tricky to match, to get the exact model. It's resource hungry, it needs GPUs, and it has long processing time. So those are sort of that's the price you pay for being able to rebase call. Not going on to the next slide. Give me 1 second. Oh. Apologies, I was on the wrong slide deck and then I lost the original set of slides, but Monika has very helpfully shared the correct set. Great. So just to wrap up very quickly, this is the this is sort of a comparison of methylation and modifications on Nanopore and PacBio. And again, that's a great feature of both of those technologies, that you can sort of keep accessing that data, and as new models come about, you can reanalyze that data and get more information out of your sequencing. And the last thing, I'm going to skip over PORC. Just know that we can replace the HiFi data that from the first slide of the HPRC sort of recipe with PORC that can be done on the Promethean. So here's hopefully this wraps it up. The current recipe for HPRC of combining all the different sequencing data types takes quite a bit of effort. You have to have these 3 different types of sequencers. It's only possible by this large collaboration. And the idea is we're hoping to be able to replicate that with just one sequencer, the Promethean, and be able to replicate that recipe with just with the different data types that we were discussing. And with that, if anybody has any questions now, we can briefly do questions, or I'll hand it over to Monika.
Thank you, Ivo, for a wonderful presentation. Yep. Just like Ivo said, are there any questions?
Okay, then I guess I will take over. So thank you so much, Ivo, for that wonderful introduction to the sequencing technologies. So the goal of this webinar is sort of for all of us to get on the same page, so that we have, the same knowledge base in terms of sequencing technologies. And of course, feel free to reach out, feel free to ask questions, or even email us after the webinar. We're happy to help. And so for the second part of the talk, I would just start by giving credit to all of these wonderful people: Julian, Lucas, Arangri, Karen, Miga, and Sergey Koren, because throughout the presentation, we used, some of their slides, some of their ideas, and I just want to credit the work they put into developing all of this. So for today, we already covered all the sequencing technologies, and I would like to focus on algorithms for near T2T genomes and on the assembly QC today. And so with that, let's get started. So first, let's start with the motivation, why do we even want to assemble the genome? And I would argue that the reason is that the gaps in assemblies are actually gaps in our knowledge. And so this is the view of the human genome before the latest T2T release of CHM13. And you can see that all of these red stretches are basically gaps. And for example, for the Y chromosome, you can see that half of the Y chromosome is completely missing. And so that's not true anymore. So we finally have a complete Y chromosome, which was just recently released by the T2T consortium. And so finally, we can get better view of these difficult regions. And not only that, but we also have a better representation of regions that are present in the human reference, but are complex. And some of these regions include, gene families with variable copy number, or genes related to the immunity. And so I would argue that it's just very important to get it right. This is a figure showing how those gaps look like. So for example, if we have the chromosome on the left, and all these colors represent the individual parts of that chromosome, you can see that previously we had gaps, we had errors, and once we can fill those gaps with long reads or correct those copy number errors, we get complete genome. So that's just one genome. But we know that when we look across the human diversity all around the world, there will be lots of variation. And here in Human Pangenome Reference Consortium, the goal is to like uncover that variation, which is also like why this collaboration with you guys and this workshop, it's so important for us that we can uncover this variation that is currently like completely missing from the databases. And in terms of like long reads and how to get there, I just want to like show this like figure where we have reference with like various differences. And when we map our reads back to that reference, like sometimes we're successful and sometimes we're not. And so with short reads, what often happens in these difficult regions is that you will get reads that are multi-mapping. And so in this figure, these like blue reads would be the ones that you were able to like map perfectly, uniquely, and you're confident that you map them to the place where they originated from. But for those, yellow reads, they will map to multiple places, or you're not really sure. And many of the pipelines currently just filter those reads out. And if you're doing an assembly, then of course these reads like won't be very useful. But luckily, if we have accurate long reads, we can span that, and the ambiguity is no longer there. So like longer the reads, the easier it is to span to these regions. And in terms of like accurate long reads, as Eva mentioned, we have a choice of like HiFi reads, or we can use duplex reads from the ONT, and we will, anchor those reads correctly. But we also have ultra long reads now, which can be extremely long. You're looking into like sometimes like hundreds of kilobases, or you're looking for reads that are longer than like 1 MB. And so you can imagine that like with such a read, even if the accuracy is lower, you can span and put together like really long regions. So why do good assemblies matter? So I mentioned that there are difficult regions in the human genome. So this is a figure from a publication by David Porobsky. And what you can see is that like this is medically relevant genome in Prader-Willi syndrome. And on the left, you can see more of like how the traditional assemblies would look like. And on the right, you can see that if you use better assemblies, you just have like so much more power to discover like what's the real variation, as opposed to like having assemblies which have some like collapses or like false duplications and errors. So how do we get there? Okay, so we want to capture the human diversity. We want to have as good assemblies as possible. And so I would argue that currently the assemblers of choice for the human genome are either Verco or HiFiasm. So those in our hands like produce the best results. And in order to use them, what you need is several types of technology. So of course you need to start with reads like, for example, that are long and accurate at the same time. For example, the PacBio HiFi reads. Then you just do some trimming and filtering process, and you will get to your like final PacBio HiFi reads. Then you also these ultra long reads, sort of to like thread it all together. And for that, you can use Nanopore ultra long reads. Depending on your data set, sometimes we would filter only reads that and keep only reads that are longer than 100 KB. And then if you're interested in like phase assemblies, meaning that like we want to fully assemble like maternal or paternal chromosome, then what you would go for is some sort of like markers for phasing, and those can be like HiC data or maybe Illumina data from the parents and other data sets as well. And after you have your assemblies, you can phase them, and then you can run several metrics on them, and we're going to cover those metrics today. So we can cover the contiguity of your assembly, how correct it is, and also completeness. And so we'll get to that in just a little bit. So first, let's cover what is phasing. So you can imagine that like if you build your assembly, you can get this like assembly graph where in the two haplotypes, some of the regions will be shared. And that just means that the sequence is like identical between the two haplotypes. But you will have also these sort of like bubbles where you might have, like maternal sequence or paternal sequence, but you don't really know like which one is which one. And then sometimes you will get like more information just by aligning ultra long reads. So you can imagine that like this ultra long read basically tells you that this part of the bubble and this part of the bubble, like lie together, are in close proximity. Or, for example, you can have some like k-mers from parents that are just like unique to those parents. And so then you can color them like blue or red. So that also gives you an additional information. So phasing just means that like we're moving away from that model of like reference genome being a single sequence, but we want to have like a diploid genome, which means that like for every chromosome, it will be represented by two sequences. Like we will have like maternal chromosome and paternal chromosome. Any questions about that? Like feel free to, type in the chat or ask directly. And so if you go from Verco unphased, you can phase with parental k-mers, you can phase with HiFi data, or as Ivo correctly mentioned, like if you have access to ONT platform, then you can do PRC data, which is basically just like a type of HiFi data or like equivalent of HiFi data for the ONT platform, or you can use strand-seq data, and strand-seq is a technology that is increasingly used for detection of inversion, and the reason for that is that it will give you information about like which strand of DNA this read is coming from, like whether it's like 5'3" or like 3'5". So traditionally, if you have a molecule, you have no idea whether it's whether the read is like forward read or like reverse complement of the reference that you're looking at. And so with strand-seq, you will get that information. And so the recipe is to have accurate reads, ultra long reads, and also some phasing markers. So that's the most common assembly recipe. And on that note, I would like to briefly introduce the algorithm of Verco, because it introduces like few steps that I think are very nice. So for example, at the beginning, when you start with your reads, what you do is the step called compression. And so you can imagine that different sequencing technologies have different biases and different like problems. And one common problem that we have are homopolymer runs. So if you have stretches of A's, basically for every technology, it's very difficult to tell you whether there's like 7 A's or 8 A's or 6 A's. But when you're aligning reads together, like maybe it doesn't matter. You just want to know if it matches. And so you can imagine that like you could compress these stretches of T's into just a single T. And then you could also do something called microsatellite compression, where like TA sequence you would compress into a single TA. And then with these reads, you can work like the work with them will be much easier. And so you will build your graph, and then you will thread it with ultra long reads. And that's how you will find out that like this blue piece and this blue piece like belong together, because like you found an ultra long nanopore read that gave you that information. And then you have this like nice assembly, and what you will do next is the phasing step. So you will find like maternal path and paternal path, and then you will get maternal counting and paternal counting. So this is the algorithm for Verco. I won't get into details of algorithm for HiFi. So that's the second assembler that we're gonna cover, but I will just say that it comes in two flavors, HiFi and then HiFi ONT. So even if you have only HiFi reads, you can run it. And even if you don't have any phasing markers, you can still run it. So I would say that like, especially for like low coverage data and where you don't have access to like big computational resources, like HiFi would probably be your first choice. If you have, tons of data and you're interested in T2T chromosomes, then you might be like more leaning like towards trying out Verco first, or you can try them both and see like what works better for you. And so this slide comes from Julian Lucas, and what I want to show here is that if you just focus on T2T contigs, and by T2T contigs I mean that like you really assemble the full chromosome. So you have telomere on one end, telomere on the other hand, and the full chromosome is yours. And in blue you can see contigs assembled by HiFi, and in red contigs assembled by Verco. And you can see that they differ. So even with exactly the same data, just by using different assemblers, you get slightly different results. And there are some interesting patterns. Like for example, HiFi really likes chromosome 12 for some reason. And then there are also chromosomes that are just challenging across the board. And so acrocentric chromosomes, 13, 14, 15, 21, 22. Those have, high copy number of like rDNA copies, like ribosomal DNA, and those are very challenging to assembly, which means that like your assembly will typically break in those regions, and so you will never get just a single sequence for them as of right now. Here is another example of combination of the technologies. So the assembly on top was generated only with the duplex data, and it has 4 breaks, and the breaks are annotated. So it's a breaking in the telomeric sequence around the centromere. There is also like some satellite DNA. There is another assembly in blue, which was generated only with HiFi reads, also has some breaks, including like this segmental duplication here. And for this case, like if you build an assembly that combines both of these technologies, like you will get like full T2T chromosome. And so what I want to argue with this slide, that like not only like different assemblers can take you to different places, but also like the combination of technologies. Like for example, PacBio reads will have a hard time sequencing through like GA-rich regions, so we will have like drops in coverage. With ONT reads, like there will be different biases. Like we, for example, know that ONT has like strand bias for certain like k-mers, where you will get like more coverage of like one k-mer versus the other. And so if you have access and you're able to, then I would argue that it's always useful to use like orthogonal technologies and see like how far they can take you. Also filtering of the data matters. And so in this case, I'm showing 2 assemblies. The one on the top assembled 19 T2T chromosomes. The one on the bottom assembled 23 T2T chromosomes. But the main difference is the filtering of the input reads. And so on the top, you can see that I use like 118 gigabase pairs of the data. But then those were just like raw data, how they come out of those sequences. But when I filtered them and like removed all the reads under quality of 20 and all the reads shorter than 15 KB, I got like much less of data. Like I only got 67% of the data, but the assemblies got better. And so that's just a, to sort of showcase that like even with input reads, the way you treat those raw reads and how you filter them and how you quality screen them, that will also make a difference. So, so it's not only about the algorithms, but also how you prepare your data beforehand. So how much input data do I need? This really depends like who you ask for. And so everyone, will give you different guidance, but also depending on like what you want. Like if you really want to have like T2T chromosome and really go telomere to telomere, then you will probably need like to have a way more data than if you just want to assemble the easy parts of the human genome. So if the easy parts of the human genome, you can probably get away with like something like 40x of HiFi, maybe 15x of ultra long, and maybe 30x of some high-C data for phasing. If you want to go T2T, then you probably want to have at least like 40x of all these technologies. So, depending on how much you're willing to invest in your data, it will take you to different places. So in summary, in order to do T2T genome deployed assemblies, we require a combination of highly accurate data and ultra-long data. And that increased coverage or increased read length will improve T2T assemblies. And so there are some trade-offs, like for example, like Ivo showed you that with the duplex data, yes, you get more accurate ONT reads, but you will also get less throughput because if each read is a consensus of 2 reads, that automatically takes you to the half of the reads that you would otherwise have. The combination of technologies can improve T2T assemblies. The example that I showed was like PacBio HiFi and anti-duplex. And the last point is that like if you only have access to ONT, like then you can still do everything on one platform and get some high quality chromosome assemblies. Okay, so we generated assembly and now let's QC it. Like let's see like how good that assembly is. And in order to do that, like you will typically start with like two questions. The first one is like, how continuous is your assembly? Like how many, pieces each chromosome is like broken into. And the second is like how accurate it is. And for that, I would argue that there are like two types of accuracy. The first one is like base accuracy, just like if I look at any nucleotide in my assembly, like how confident I am that that nucleotide is correct. But then there's also like structural accuracy, meaning like, did I assemble like correctly these complex regions? Like, is my, copy number of genes correct? Like, did I accidentally like join like two different contigs that like should not have been joined? And so all of these things matter, and there will also be some, trade-offs that sometimes maybe if you're willing to have your assembly like a little bit more fragmented, then you will make less mistakes in like joining them together. But then also if you want to analyze your genome as a whole, then maybe you want to have like full chromosomes even at the cost of like making a few mistakes. And so this is just figure of assembly continuity. The traditional metrics are like N50, where you basically take the length, half of your length of your assembly, and you look like, okay, what's the length of that contig? You can also ask this question in terms of like the genome size, and you can say like, okay, how many contigs do I need to assemble half of my genome? And you know the genome size, like if you assemble a human genome, then you know that you should expect to get around like 3.2 like times 10 to the power of 9 base pairs, right? So you can ask all those questions. Or if you're aiming for like T2T assemblies, then you can ask like, okay, how many T2T chromosomes am I getting? And so that's what you can see at the bottom, where you can see on the x-axis all the chromosomes, the maternal and paternal haplotypes as 2 rows. And whenever you assemble the chromosome fully, telomere to telomere, you will mark it in a green square. And when there are some like gaps, visually we only see like handful of gaps, like few kb, or maybe at most like few dozens of kb, then you will call it like T2T scaffold. And so this can be like another metric. So again, like it depends on your goal and what type of assembly you're aiming for. The next metric is a Q value estimate. And Eva already like showed you how these probabilities look like. And I would also like to recommend the Q100 project with Adam Filippi's group, where they're focusing on the AG002 individual and trying to get like absolutely the best assembly possible with the technology that we have now available. And also in terms of phasing and getting like maternal and paternal chromosome, we are trying to get like red chromosome and like blue chromosome, but like of course like things don't always go well, and we can make different types of errors. And so for example, here I just want to introduce switch error and Hamming error, where imagine that like this contig should only be red. It should only have like red markers. But you can see that like probably, two contigs got joined together that should not have been joined, or something like that happened. And you can see that from this like red, you're switching to the blue. And so that means that your switch error will increase. And so in this case, the switch error will be 11%. Another metric you could use is like Hamming error and just say, okay, this contig is supposed to be like red, but I see some blue. So the Hamming error rate would be 20%. And so these two metrics will be also useful in evaluating your assemblies. The last I want to introduce is something called completeness. And in this case, we often use a tool called ISM Gene. And the idea here is that like when you have your, perfect assembly that you put lots of effort in and you're very happy about that assembly, then imagine that you will like align it to AG38, or you will align it to CHM13. Then, depending on like which tool you use, like for example, you could use like Quast or another tool, like you will find like misassemblies, like you will find differences between your assembly and the reference. And then the question is like, is my assembly wrong? Maybe some parts of it wrong, but like no two individuals are going to be the same. So unless you're assembling the individual, which already has extremely high quality reference, you do expect differences between your individual and the reference. And we, expect differences in SNPs, of course, like just the diversity that you would expect in human populations. But there will also be like large amount of like structural variation, which are also like expected and normal. And so maybe one ways to go like around this issue is to look at something that we do expect to be like more or less conserved. And so in this quality metric, what you're asking is if the single copy genes that you know are in the human reference, is there are still single copy in the assembly, and if the multi-copy genes that you would expect to find in the reference, if there's still multi-copy in the assembly. And so just in these schematics, you can see that this blue transcript is present twice in the CHM13, but in your new assembly, it's no longer multi-copy. And so that could be an indication that your assembly is like wrong and it collapsed the gene. And so with that, I would like to thank you all for your attention. I would like to credit all people that contributed to the slides. I would like to especially thank Benedict Patton and Karen Miga from UC Santa Cruz Genomics Institute. And I think we still have a few minutes maybe left, so we can still answer some questions, and both me and Eva will see you in person next week. So we're very excited to talk to all of you. Thank you.
Thank you, Monika, for such a lovely presentation. Are there any questions from the attendees?
I think everyone is exhausted after 3 days of webinars.
Well, probably. I don't see any questions. Can you just quickly check the chat box? Oh, so Hussein has a question on the chat box. How can an assembly be annotated?
Yeah, so it depends like which kind of annotation. So in terms of like annotation, for example, of genes, this is not my area of expertise, but like what people generally use is that they either like use transcripts and annotate the assembly like de novo, or often when you have your new assembly, you try to do sort of like one-on-one match to an existing assembly and do like something similar to like liftover, that like you already have this like well curated like genes, like can you find them in your assembly? Then, you know, in terms of like annotating like centromeres, telomeres, like repeats for those like specific pipelines that include repeat masker. We also like in Miga Lab, we have pipelines to annotate like centromere and how they look like. Yeah, so really depends. Like it's either always like de novo or using annotations from a reference. So that's why one of the like main pushback actually in using like CHM13 and like all these like complete reference genomes is that there's such a, you know, like wealth of annotation for like AG38 or even like AG19 that like lots of people are like reluctant to, you know, like go one step and like start using these new references just because they're worried that they will be missing some annotation data. But every metric that you look at, including, you know, gene copy number, accuracy of like variant calls, I would argue that like the complete assemblies like do matter and that you will get better results.
Any other question? Well, I don't see any other question. I don't know if probably everyone understood what you guys presented today, and they've benefited quite a lot from it. And I also enjoyed the presentation as well. So since we don't have any other question from the attendees, I guess we can end the session here. Thank you, Ivo and Monika, for gracing us with your presence and presenting to us about the pen genomes and the various tools that we can use. And thank you everyone for attending this webinar. See you next week for the RefGraph workshop. Bye.
Thank you. See you all. Bye-bye.
Thank you. Bye.